Biostage is a small, publicly traded biotechnology company located in the greater Boston area with a focus on the development of novel combination products for the treatment in the field of regenerative medicine. Today I'm going to tell you about our lead product candidate, which is the Cellspan Esophageal Implant. The Cellspan Esophageal Implant is a combination product. It is a cell delivery device as well as a biologic. The direct delivery of autologous adipose-derived mesenchymal stem cells to the esophageal conduit through the production of a modified polyurethane tubular scaffold, which we term the cell frame scaffold, provides temporary support during the initial tissue regeneration phase. The cell frame scaffold is a temporary device. It's removed approximately 21 days after implantation and leaves behind the cells and new tissue growth. The uses for this product are for segmental esophageal reconstruction, for end-stage esophageal disease, as well as for the congenital defect known as pediatric atresia. This is an incomplete esophageal tube that occurs in about 1 in 3,000 children. The function of the CEI is to induce host tissue growth and to provide us and to stimulate constructive wound healing within that tissue, to induce by laying down a permissive extracellular matrix as well as inducing angiogenesis. This bubble diagram just indicates all the steps required when you need to manufacture a combination product. The cell scaffold or the cell frame scaffold is produced in-house. We have an ISO-compliant materials manufacturing suite. The scaffold is electrospun. In addition to spinning that tubular scaffold, we need to do all the characterization in-house. The second phase of this is to actually characterize all the cell component or the biological component. And then finally, putting those two components together to make that final combination product, you need to characterize what that final product looks like. This is just a diagram showing you the manufacturing process for the cell frame cell delivery device. Again, it's electrospun, it's polyurethane. We can make various sizes depending on the size of the organ. And again, these are tubular structures, and our focus is on tubular organs. We characterize the pore size, the fiber diameter, as well as the tensile strength, pre and post implantation, to demonstrate that the scaffold itself is stronger than native tissue. In addition, we do a surface plasma treatment in order for the scaffold to become wettable and allows for cell adhesion. Prior to the plasma treatment, the polyurethane scaffold repels water and cells do not adhere. Post-plasma treatment, we get water absorption and cell adherence. Well, we next need to define what that drug substance is. We've spent a lot of time looking at different cell types. We focused on the adipose-derived MSC. These are vascular-associated cells taken from adipose tissue biopsies. The cells are isolated via enzymatic digestion, grown and expanded in culture, and characterized down to the molecular level. The cells do conform to ISCT standards. They express CD73, CD105, and CD90. In addition, we've characterized the secretome, the growth factors, and the cytokines that these cells express. We've also done proteomic analysis to try to pinpoint the mechanism of action. Again, the cells express cytokines and primarily express cytokines that are involved in angiogenesis. VEGF, MMP2, IL-8. We've done some in vitro angiogenesis or vasculogenesis assays where we take human vascular endothelial cells, umbilical cord vascular endothelial cells. We can incubate those cells with conditioned media from the AD-MSCs and show that the HUVECs form capillary structures. In addition, we've also characterized the extracellular matrix and have defined what that extracellular matrix is composed of and characterize that as a permissive environment for for new tissue ingrowth and regeneration. The final product, the CEI final product, again, is a combination of adipose-derived, and these are autologous cells, so there's no issue with immune rejection. The cells are grown in 2D culture to expand them. They are then seeded onto the scaffold in a custom-designed bioreactor. This bioreactor, our most recent version of this bioreactor is a fully closed system. It uses an automatic seeding process. That final Cellspan Esophageal Implant is a tubular structure. It's coated with cells. The cells actually grow into the interstices of the matrix. There's a SEM indicating cell growth on those fibers. And then part of our manufacturing process, we monitor in-process controls or the metabolic activity to show viability and metabolism. We quantify the DNA content on the final product to demonstrate, the dose of the cells. We also characterize the secretome associated with the mechanism of action. This, in order to test this, we've developed a large animal model. We currently use the Yucatan mini pig porcine model. We take an adipose biopsy at day -28. We expand the cells through a standardized cell culture manufacturing process. We then seed the scaffold. The animal then undergoes a thoracotomy. We segmentally remove a portion of the native esophagus and implant the CEI. The scaffold is removed at day 21. It typically comes out on its own, but we actively go in and remove it endoscopically. We then do a series of stent exchanges while the tissue regenerates, and those stent exchanges occur over the course of the first 3 to 6 months. Following that 6-month time point, we remove the stents, the animals are stent-free, and then at various time points, we harvest that tissue. We did an acute tox biodistribution study with GFP-labeled cells, and what you'll see here is a 2-week, 3-week, and 4-week time course. Well, you see the regeneration of the epithelial cell layer and the mucosal layer of the luminal surface of the esophagus. And you'll see as time proceeds, the unepithelialized implant zone becomes smaller and smaller, and we've done morphometric analyses of this. Typically what happens is we get this fibrovascular tissue regeneration in the early phases, as you'll see from the H&E staining in the top panel. We can define the implant zone based upon the histology. The other aspect of what the cells do is they induce angiogenesis, and you'll see in the green cells surrounding new vessel formation within that implant zone. And this actually provides blood supply and oxygen to that implant area, and then we have never seen a necrotic event associated with the implantation of this. The time course of regeneration, as I said, we did an early acute phase biodistribution and tox analysis from 30 days through 270 days through animals that have survived out past 600 days. And what you'll see is a progression of the regeneration response. The first thing we see is reepithelialization of the lumen and the mucosal layer. We then see the muscularis mucosae develop over time. And then we begin to see the tunica muscularis segment of the esophagus develop with smooth muscle cell migration into that implant zone. And this is very characteristic in all the animals that we've done. This is a typical regeneration process that we see. So the target product profile for this CEI product, combination product, for adult indications, it's end-stage esophageal disease, early-stage cancer, intractable strictures, non-healing chronic recurring fistulas, either caustic burns and or injury. A primary focus of Biostage is to apply this product into esophageal atresia, and again, this is a congenital defect that occurs in about 1 in 3,000 births. These patients are born without a fully complete esophagus, requires surgical intervention. We believe that this product will be ideal for that situation. Back in 2017, the Mayo Clinic had a patient, and we had collaborated with Dennis Weigel in the past, head of thoracic surgery at the Mayo Clinic. Had a patient that came in. He was a 75-year-old male. He had non-small cell lung cancer. The cancer was growing in his chest. It impinged on the heart, the vena cava, as well as on the lungs. The patient wanted to be part of an experiment for our new product and volunteered for an emergency IND compassionate use application of the Biostage CEI product. The surgical procedure for the implantation required a patch on his heart, cardiopulmonary bypass, a Gore-Tex graft on the vena cava, as well as removing a large portion of the lung. The tumor itself impinged on the esophagus. 4-centimeter segment of the esophagus was removed, and 5 centimeters of the Biostage CEI was implanted into this patient. The patient survived for 7.5 months, eventually died of a stroke. But what you'll see from this figure is that the regeneration process that we saw in the implant zone in this 75-year-old patient mimicked to what we see in any aged animal that we've implanted in the past. So we did a histological comparison of the flanking native esophagus, the transition zone, as well as the center of the implant zone. And we compared that to a 270-day porcine recipient of the CEI. And what you see is almost an identical process of regeneration, where again, we see the epithelial cell layer and the mucosal layer regenerate first. In that transition zone, we begin to see smooth muscle cell migration as indicated by smooth muscle actin staining. We see almost a complete submucosa, mucosa muscularis layer. And then as time proceeds, we actually see smooth muscle cell migrating into the implant region in the tunica media. This is just a summary of how we believe the CEI works in the healing process. The initial implant, the cell frame scaffold, as well as the CEI itself, forms very tight anastomotic ends. We believe that the MSC component of the CEI helps those anastomotic sites to heal very tightly. The cells actually deliver cytokines and growth factors to induce new tissue ingrowth. At 21 days, the scaffold is removed. The epithelial cell layer starts to migrate in and actually peels away the scaffold. We actually go in and actively remove it at 21 days. And then the cells continue to stimulate vascular genesis and neogenesis of new tissue. In summary, we've done a whole series of proof of concept and GLP preclinical studies that demonstrates that the CEI directs esophageal repair following a full thickness resection. We get new tissue ingrowth. We get luminal epithelial migration and mucosal regeneration within 90 days. Angiogenesis is stimulated within the first 2 weeks. We end up with a functional biological conduit, and we have long-term surviving animals up over 600 days. We've completed the EIND compassionate use case. Again, the gentleman survived 7.5 months. This was recently presented at the STS TechCon this year. And we've observed that the regeneration process is almost identical to what we see in the animal model. So this is actually demonstration that the process of regeneration that we see in animals occurs in the same way in humans. This is a platform technology where we can tailor make any tubular organ and seed it with any type of cell to regenerate damaged tissue and cells. And where we are today is we're in the process of filing our first IND. just to acknowledge all our collaborators, the Biostage team, our collaborators at the UT Health. We use UT Health GMP facilities to manufacture final product. We use them for the EIND, and we're currently using them to move forward for our first clinical study. Dennis Weigel at the Mayo Clinic, our animal CROs, our collaboration with Connecticut Children's Hospital, and the receipt of a $1.7 million grant from NICHD with a SBIR fast track, and then our regulatory consultants, Boston Biomedical Associates. Thank you very much.